Hello?
Hi.
Hi, how are you?
Oh, pretty good. It didn't look like you. This is 14, meeting 14. Is that right?
I think so. Why?
Okay. Yeah, I couldn't find it in my email.
Oh, I don't think I sent an email.
Oh, okay.
Yeah, I think I'm gonna just do a check-in today because we have construction in the house, so I don't wanna.
Oh,
Yeah,
oh my. That's always fun.
you don't wanna. Yeah.
Yeah. Okay. Well, I managed to figure out how to get a stress and strain um, table out of console.
Oh, good.
I asked ChatGPT. that, that was a recommendation of the grad students I'm associating with,
Oh,
like
great.
their a D-bio.
Yeah.
And so I did ChatGPT, and they told me to put a probe in. So I put a probe in the spot that I've been trying to get the stress strain curve of,
Yeah.
and it worked. I have a table. I still don't have a graph because I don't know how to do graphing.
Well, can't you just graph the tables using something else? It's fine.
Yeah, well, yeah, I can. I threw it in Excel.
Yeah.
I give up.
That's good. Yeah. Well, you got what you needed. That's the way it goes.
Yeah,
That's
the
great.
table is fine. Thank you. I can put that in Excel and then throw it in MATLAB and do whatever I want with it. But yeah, put a probe in, and you find the probe here.
Oh, okay.
So if you're ever having trouble, well, actually, they recommend it for programming.
Yeah, they do.
If you're having trouble with your code, put it in ChatGPT.
Yeah.
Well, finally, we may have reached the era where computers do what you intend instead of what you tell them.
Yeah.
Oh, yes. Yeah.
So, yeah, we got our reviews back from the paper on the Life special issue, and there was some discussion in the emails and things. So, what's your opinion?
My opinion.
Yeah.
I think I sent you the last one, which is I'm game for a journal if you're prepared to go through another round of reviews. and the only thing is, I'd like it a journal that doesn't cost us an arm and a leg.
Yeah, I know.
And which allows reproduction by the authors.
Yeah.
Okay. There are lots of journals. I gave you 2 examples, but there's lots of astrobiology journals now.
All right.
Okay. If you have any problem with journals, I can try to work up a list.
All right.
But basically, when I collaborate with somebody at my age, the choice of journal has no effect on my career.
Right.
Okay. So
Yeah.
pick a journal that's useful to you.
Yeah, it's hard to find something that's like, well, sometimes they don't charge APC fees, but that's always like, I've had that problem before where like.
Yeah, there's some journals that charge only if you want to pay for open access.
Right. Yeah.
They're free otherwise.
Yeah.
Okay. So you have to read the fine print.
Oh yeah, definitely.
But this journal it's a Chinese journal, this Life thing.
Yeah.
They're imitating the same name as a journal called Life from Switzerland.
Oh really?
Yeah, it's the same name.
Yeah.
And they obviously, I just did a review for a journal in Serbia, and they very clearly state rank this paper by acceptable as is, with minor revisions, with major revisions, or reject.
Right.
And they very clearly give you the options.
Yeah.
But this journal gave no options, so they left it completely up to themselves.
Yeah.
And so if they interpret anything negative at all, they can say, "Oh, you got a negative report."
Yeah.
As I said, it was a game that the grant agencies used to play. if they didn't want to give you any money, they would send out to 11 reviewers to take the worst one.
Oh, right. Yeah. So well, we'll just find the one that differentiates it from good.
Yeah.
And
Well, you know, the more reviewers you have, the more the probability is of getting a bad review.
yeah, that's true.
And I'd say some of the reviews we got were very good. So a good editor would say, gee, I'll go with this because I got some good reviews.
Yeah.
So it's obvious that they're in it for the money.
Well, yeah. But yeah, I thought there were some good reviews. I mean, they had a couple good points about
Oh yes, I'm not
getting
arguing.
the, yeah.
I'm arguing with the journal.
Oh, yeah, I know.
There was a Revex journal that's, but it's biomedical engineering. They've got a new journal, and they say they're going to have it for free for a year. All the fees.
What is this?
RIVX. I don't know.
Oh,
I
I have to see.
There's a group of journals.
Yeah, obviously Bashir's a physicist. He doesn't care where it goes.
Yeah.
Okay.
Yeah.
And your contribution is much bigger than his.
Yeah, I know. I think he just did some equations or something.
Yeah, he did. Yeah, basically he based it on taking a stat next course.
Oh, okay.
Okay. Which is fine.
Yeah.
I don't know where that went. It was from the physics conference.
Oh,
Anyway,
okay.
one of the APS type physics journals. Oh, maybe this is it. PRX Life.
Oh, yeah, I know what that is.
Sorry, yeah. And they've got the biophysics is free for. There's one of them that's new and it's free for a year.
Yeah, I think we have to emphasize that we're doing toy models. We
Yeah.
have to publish it someplace that gets the idea of toy models.
Yeah.
I mean, I attended a lecture in Washington, D.C. In 2015 by a young lady who lectured about 100 definitions of life.
Okay. Yeah.
Oh my goodness.
Okay, and I'm sure the number's gone up since then.
Yeah.
Okay, so we're just taking a very naive definition. If it works, it's alive.
Right.
My actual definition of life is very simple. If it can eat you, it's alive.
An amoeba.
Or if it can kill you, it's alive.
Well, okay, yeah.
Okay.
That's interesting. Yeah.
So that's based on watching Jurassic Park.
Right. Yeah.
Okay. So
All right.
how you doing, Jesse?
He's usually not able to speak on Monday mornings, but
Oh.
well, he can't turn on his
What does
audio.
he eat on weekends?
Oh, yeah. Yeah. So yeah, I'm not going to do a full meeting. Oh, here he is. Yeah, he's recovering from a lot of travel, and he had a conference that he did this last weekend. So he's been busy.
No longer my problem.
Yeah. Well, I'm not going to do a full meeting today because we're doing construction in the house. So I expect them to be drilling and things. So.
Yeah, I attended a chemistry lecture once where there's some guys using hydraulic pumps to break concrete just outside the hole. So they were asked to stop until 5 o'clock, and right at 5 o'clock they started up again.
Yeah. Yeah, I can't remember what else I was going to ask. Yeah, so Oh yeah, I was gonna say about the Google Summer of Code projects. So those have been in, so they've been turned in, and we're waiting for our slot decisions to be made. So we may have one or two people working on it this summer, hopefully in that project, that the Graph Neural Networks project. And I also had some interest from Hari Krishna on revitalizing the digital microsphere stuff that he was doing last year. So that was, I don't know what he wants to do in that, but yeah.
Well, now that I've actually got some data, maybe I'll get this project done and I can take a bit of a breather and get pictures from my microscope.
Yeah.
That would be nice.
Yeah.
Even if it's shot, those paintball shot balls.
Right.
Stripes. I don't know. Yeah. I just, yeah.
Yeah. It's, anyways, just to let you know where that stands. So yeah, I hope we can get some good work done this summer. A lot of contributions to the web, to the our DevoWorm IO site that I showed last week. They've been Sushmant and Jyothi have been contributing to that quite a bit. So they've been making a lot of pull requests, putting things in. So it's a lot different now, and they'll be. I guess they're going to be putting up like a blog infrastructure on there, so we can have we'll use the blog entries from some of the previous projects as documentation, and then they're going to be putting in blog posts this summer, or at least Sushmant is. I expect Sushmant to get accepted, probably, and he wants to do this for like his master's project, I think, too. So he's got a whole plan. So definitely we're gonna at least have Sushmant with us this summer.
Donald Ingber has a new paper out, 2023.
Oh yeah.
And it's a good reference. Like it's sort of a summary of his work on tensegrity, so it's kind of a good reference. And what's the other thing? Oh, I downloaded Morpho.
Okay.
So we'll see if I'm going to see if it works. I'm in contact with one of the people who made it, Timothy Albert. Oh dear, starts with an A. He's a professor at Tufts University in soft matter physics.
Right.
All right, Susan, got a question about Ingber. Did he ever get past tensegrity with fixed topology? In other words, where the topology of connections can change.
I don't know. I don't think so, but I don't know. It's the form finding tensegrity is that's that topic.
Okay, yeah, I guess about 13 years ago I met with, what's his name, Staminovich, guy who works with him.
Oh.
And I think that seemed to be where they still are at.
Well, and I've tried to find papers about tissue and tensegrity of tissue and so on, and they keep taking a single cell and saying, oh, this is an epithelial cell and we're going to work with this, and it ends up being like an amoeboid cell or something.
An isolated cell.
Yeah, so I still think I've added something to the model with my model. So
good.
Yeah.
So I think it's worthwhile doing for just my candidacy project.
Well, Susan, consider the possibility of a sphere with a small pressure. so that you eliminate the edge boundaries.
Edge boundaries?
In other words, if you have a sphere, there is no edge to the sheet of cells.
Well, there is no, my workup, there is no edge really. It's like everything depends on its neighbor. Like they all.
Yeah, but what about the ones at the edges of the piece of the sheet that you use?
Well, they have to be tied down somehow.
That's what I mean. By putting it in a sphere, you don't have to do that.
Oh, I see. You want to do it on a sphere. Yeah.
Yeah.
I hear you.
Okay.
I hear you. I don't know when I might do that, but it is an idea. Yes, thank you.
Okay.
Yeah, and that reminds me.
There's a very small bit of literature on the size of the pressure inside an egg, which would retain the spherical shape.
Well, I think that's the whole thing with an amphibian egg is there's actually a pressure in there.
Yeah, I recall that Beluso had in one of his books, I think he estimated at 5 atmospheres, which is ridiculously high.
Well, I've had them explode and go.
Oh, you have?
Yeah, I've had them explode on me, and they do go about that far. So that's why I came up with the idea of using a egg cannon and putting it at 45 degrees and then poking it and having it shoot off and then finding out where it landed, and then I measure the pressure because it's just an egg cannon.
Yeah, Bradley, you probably don't know this, but the CBC, Canadian Broadcasting Corporation, used to have a very funny team that included a chicken cannon, It was a rubber chicken cannon.
Rubber chicken. And there's another paper, and the researchers reduced the amount of ATP inside a cell, and it made it it's stiffer. So the cytoskeleton is no longer the dominant mechanics in the cell. It's a cytosol. It's got ADP in it. So I'm going, oh, well, there's my quote from Alive to Dead. I've been saying that, like, it changes. If your cell is alive, it's got a different mechanics from a dead cell. Well, people know this. But
unless it's a zombie.
Right. Zombies know how to use ADP to run around. That's why they're stiffer.
Right.
Oh dear, I was trying to find it and send it to you. It's a good quote, because it's the only one I've found that even gets into rigor mortis.
Okay, we should let Bradley get started before the jackhammer starts.
Oh yeah, okay. I'll just see if I can find it.
So yeah, I guess I could start here. So yeah, I'll just share my screen. So this is the stuff for DevoWorm.github.io I was mentioning. We have a number of pull requests here that have been, well, I think Sushmant has merged them. But I've just reviewed them. So they're probably about 10 from this week, maybe. And it's just adding the blogs page, changing some of the aspects of that. So there's a whole sort of documentation section in there. And so that's nice. Thank you for those contributions, Jyothi and Sushmant. Okay, Sushmant ready and Jyothi Swarup ready. Then I wanted to point out that our paper in the new issue of Royal Society Interface Focus is up. So we were working on this paper over kind of November and December. I sent or we talked about it in the meeting. So this has been published now. So this is, yeah. So this is the paper here, or this is a special issue. So this was organized by Michael Levin and Adam Saffron and a couple other people. Jesse and I know Adam Saffron, and of course we know Michael Levin. The cover image of this, let's see if I can pull this up. The cover image was actually made using AI. So Mike Levin prompted Midjourney, which is an AI program, to produce this figure. Yeah. So this is basically, he used it to describe the scaling of goals from cellular to anatomical homeostasis and evolutionary simulation, experiment, and analysis. So that's what that looks like. And it just generates an image from what it's been trained on, which is a lot of the internet. So it's like some sort of chicken person and a tree. So
it's like an old-fashioned Darwinian tree.
Yeah. So the special issue is really good. It has a lot of interesting things. They talk about the theme making and breaking symmetries in mind and life. And there are a lot of papers on like things like active inference, and there are a couple things on early life. So Stuart Kaufman published 2 papers in this issue. So one is on RNA autocatalytic sets, and the other one is a third transition in science. And both of those are, I think this one's open, this one's not. So I don't know. I've been having trouble getting the articles out of this issue because the library access that I have doesn't allow you to get papers for this from the last year.
Bradley, I've got access to 2 libraries different from yours.
Okay.
So if you want to send me the list, and indicate which ones you need.
Okay. Yeah, that
Okay.
sounds good. All right. Yeah, we'll try it. Thanks. Then this is our paper here. So.
Oh, we got in there.
Yeah. So there's our paper, and it's in the collection. Who else is in here? Who's really notable? David Krakauer, who works at Santa Fe Institute. Who else?
Did any of them cover symmetry breaking and origin of life?
Let's see. I don't think origin of life, but definitely. Well, the Stuart Kaufman article is origin of life, but this is symmetry breaking is maybe. Maybe this one here, David Krakauer, symmetry, simplicity, broken symmetry, complexity. So yeah. Oh yeah, it cites Herman Weil from book Symmetry, and Philip Anderson, More is Different. So this is.
There's a curious thing which I just ran across a couple of days ago. The possibility of breaking, of a racemic mixture breaking symmetry and forming subparts that are each of which has an antiomorphic excess, I guess it's called. Right? And I was thinking back, that's how Pasteur originally discovered things, because he had a racemic mixture of tartaric acid, but he had these tiny crystals, and the crystals themselves were polarized. And so when he separated them by hand, he was able to demonstrate polarity.
Okay.
So that may be related to this. It's symmetry breaking of mirror symmetry or something like that. A guy named Ribbo, R-I-B-O, and he's written a lot of papers on it.
All right.
Okay.
Sounds great. Yeah.
Most of them in the context of origin of life.
Right. Yeah. So, yeah.
If
The
you want, a list of his papers, I can send it to you.
okay. Yeah, that'd be good. That
Okay.
would be good. Let's see. So we have yeah. There's some interesting. There's this one. Chiral conformity emerges from the least time free energy consumption. So this is the chirality of biological polymers, and this is at the origin of life. So there is a fair amount of origin of life in here, actually.
Oh, okay, good.
So yeah, the predominance of matter over antimatter is presumed to follow some subtle bias for matter at the dawn of the universe, and then talking about handedness and chirality, and then free energy minimization. So that's good. And then this paper here, which is mixed anhydrides at the intersection between peptide and RNA autocatalytic sets, evolution of biological coding. So this is where this is talking about the origin of biological coding, and coding originated from a cooperation between two originally separate, collectively autocatalytic sets, one for nucleic acids and one for peptides. Upon interaction, a series of RNA folding directed processes led to their joint cooperativity. So this is kind of goes through some of this process.
They don't do molecular dynamics, do they?
I don't think so. Yeah, it looks like they just do.
These sound like hand waving statements.
Okay, yeah. Yeah, and then yeah, then that's pretty much it. It's like 15 articles, so it's quite a nice little collection.
There's a general argument which is made by I think it's Steve Benner, that if you have any complicated organic reactions, in general, what you get is tar.
Yeah.
Okay, so to only look at one pathway leads to what you want.
Right.
It may be very delusional.
Okay.
Okay.
Yeah.
I've come up with a counter argument to them, but in a very specific case. Okay, but
Okay.
in general, have you ever seen organic chemistry?
What do you mean seen it? Like
Seen people doing it?
not really, no.
Oh yeah, well, if you make a big mixture of molecules, in general you get black tar.
Oh yeah.
Okay, that's the idea.
All right. Yeah. Yeah, that's great. So yeah, I think this is our paper. So this is the metadata here. There's nothing really to see too much there. Just to say it's part of the issue. And then it's kind of interesting because I remember when we started this paper, we were originally invited. Maybe this isn't the right one. We had it was a different set of papers that they had abstracts for. So I think they went to a different journal originally, and then they moved to this journal. And so they got different papers in the issue. Yeah, no problem. I'll do that in the Slack. So I'll put the screenshots of the papers. Actually, I'll put together a library of the papers if it can get a hand.
Who's working on the concept that embryogenesis is a sequence of symmetry breakings?
I'm not sure. I don't know if anyone in this collection is. I know I've seen papers on it, but I can't remember.
Because especially with Susan's data, might start looking at sort of standing back from the data and saying, can we regard it as a sequence of symmetry breakings?
Yeah.
Or is something else going on?
So yeah, symmetry breakings are like just like events where the symmetry ceases.
I published a paper a long time ago. I think it was centenary of Turing, showing that Turing's model does not work if you consider consecutive symmetry breaking.
Yeah.
Hi.
Okay, because the model itself is symmetric.
Right. So it relies on symmetry.
I cannot believe how many.
It works for one step, but not for two or three.
Oh, okay.
Sorry, what was, I missed that, of
You
course.
missed that. Oh, I wrote a paper a long time ago on Turing's model for morphogenesis, and showed that it works fine for one step of cell differentiation, but if you have more than one step, it collapses because there are too many ways you can break the symmetry.
Oh, okay. I found the paper I was looking for. It's called Activity-Dependent Glassy Cell Mechanics 1. Mechanical Properties Measured with Active Microreology. At least I think that's it. And where is it? ATP-dependent cells showed an increase in elastic modulus at low frequencies. They've done some experimentations.
Okay.
So I thought that was good. It was experiment-based.
Yeah.
Anyway,
Does the same thing happen with formalin?
you'll have to ask them.
Okay.
Here. Should I just send you the paper then?
Yeah, sure.
Yeah.
Oh, Jesse, do you want the paper too?
Yeah, I can send it to him, I guess.
All right. Well, I'll just.
Yeah.
All right, because I just thought it's the first time I've had a paper that actually quotes something that I've known about for a long time, that a live tissue is different from dead tissue, and everybody knows it, but where's the quote? So it's right here.
Susan, where does scar tissue fit in your spectrum?
Scar tissue?
Yeah, scar tissue.
It's just too much fibronectin.
Okay. Okay, so it's still alive, but it's
yeah, it's being
its properties towards the dead tissue, or no?
Maybe it's sort of the same as cartilage or ligament, because they don't have a blood supply like the rest of the body. So
okay, gotcha.
that they might actually have some of these traits.
So they depend on diffusion to stay alive?
Yeah, or movement. Well, they're moving around the joints.
Oh, right, okay. Right.
And so it tends to pump the synovial fluid around. But they don't have a blood supply. They have synovial fluid. So.
Okay,
Yeah.
so that could be part of the problem of being bedridden.
Yeah, I suppose, although I was reading about that. What was I reading about? It's sort of like being in space. You're not getting that feedback into your bone, and your bone is sort of like your blood vessels in a way. The endothelial cells of blood vessels change cell.
Oh, that's why they insist that astronauts do a lot of exercise.
Yeah.
If they're not, yeah. So anyways, if they're not experiencing flow, then they change cell type. There's lots of studies on endothelial vessel cells, and some new recent ones as well. I collect them. I don't know why. I have a computer full of a collection with no end note attached.
Oh, right. It's always very hard to understand when you go back 6 months later and see.
Yeah, I have them in files, but sometimes trying to find one is extremely difficult. So I have to do something about that, but I've been pressured to do my tensegrity project. So
All right, yeah.
yeah,
You have to be cloned. You just have to be cloned.
just cloned. Yeah.
Yeah. So, yeah.
And to sustain that, I just work at it 40 to 50 hours a week. I can't do more than that. Like, this is an 8-hour day, and I need to quit.
And you have a farm anyway, which must take some time.
I don't know. If you start at 9 o'clock in the morning and work until 5 at night, and you only have like a 15-minute break for lunch,
Yeah,
that's enough.
yeah.
Right, it's enough.
All right.
How productive I've been.
Yeah. We do one more thing here. Let's see. So we've been talking about origin of life, and there's this new paper, and then there's another paper in here I want to talk about. So there's this new, I guess, a new theory or something. This is
what's that? Suzanne Walker.
Sarah Walker. Yeah.
Sarah Walker.
Yeah. This is life. Modern physics can't explain it, but our new theory, which says time is fundamental, might. So this is actually a podcast, but this is the preface of the podcast, and then there's a paper associated with it.
Oh, so she's the one who gave the lecture on 100 definitions of life.
Oh, okay. That's great.
Oh,
Yeah.
wow.
So this, so over the short span of just 300 years since the invention of modern physics, we've gained a deeper understanding of how our universe works on both small and large scales. Yeah, physics is still very young, and when it comes to using it to explain life, is a struggle. So even today, we can't really explain what the difference is between a living lump of matter and a dead one. But her and her colleagues are creating a new physics of life. So the importance of time. Isaac Newton described a universe where laws never change and time is an immutable and absolute backdrop against which everything moves. Darwin, however, observed a universe where endless forms are generated, each changing features of what came before, suggesting that time should not only have a direction, but in some ways folds back on itself. This is this idea of temporal irreversibility. Of course, we know that entropy is irreversible, and they're arguing is evolution, I guess, is irreversible, which, so I've seen papers where people have made this suggestion.
Not quite true.
Well,
if you look at small systems, it is reversible in a small system.
Yeah, I mean, it's reversible, but it goes like it's part of the history of the lineage.
Yeah, the average certainly goes forward, but an individual small system, it doesn't have to.
Right.
At least for short periods of time.
Yeah. New evolutionary forms can only arise via selection on the past. And then, so presumably these two areas of science are describing the same universe. But how can two such diametrically opposite views be unified? And
let me make a comment about that last sentence.
Yeah.
There is a group of people who are searching for independent origins of life.
Yeah. Yeah.
Including in the present.
Yeah.
Okay. So that would conflict with that statement.
Right. Yeah. Yeah, I wonder, that's true. I mean, why doesn't life start up like now instead of back in early Earth? And of course...
Yeah, the general consensus is because it would be eaten right away.
Right. Okay, that's the citation Susan made from the last...
Which is not to say it doesn't happen, but it gets eaten.
Right.
Oh, I missed one part. Sorry, it's lopsided.
That's okay. So the key to understanding why life is not explainable in current physics may to reconsider our notions of time is the key difference between the universe as described by Newton and that of Darwin. Time has in fact been reinvented many times throughout the history of physics. So there's this whole area called the philosophy of time, where they talk about like how time is conceived or how to conceive of time. So in Newton's time, time was fixed and absolute, whereas in Einstein's time, time became a dimension, just like space.
Question.
Yeah.
In Einstein's view of time, is time reversible?
I'm not sure. I don't know if it is.
I don't either. I haven't studied deeply enough.
Yeah. So it's just a dimension, just like space, but whether it's reversible, yeah. And then, of course, you know, we've talked about second law of thermodynamics, where you know there's this idea that time is or at least the flow of energy is irreversible in time, and so that's another view of time, I guess. And just as all points in space exist all at once, so do all points in time. So I guess this is referring to Einstein's view of time. This philosophy of time is sometimes referred to as the block universe, where past, present, and future are equally real and exist in a static structure with no special now. In quantum mechanics, the passage of time emerges from how quantum states change from one to the next. So this kind of goes through this this talks about thermodynamics. The invention of thermodynamics gave time its arrow, explaining why it's moving forward rather than backwards. So all points in time exist, but you're moving in one direction. And so, you know, anything in the universe that's operating like as a dynamical system, I guess, such as a working engine, that's irreversible because it only works in one direction forward. And same would hold true for life and living things. Then, you know, to go directly to evolution in life, specific to that, to build novel things, evolution requires time. Endless novelty can only come to be in a universe where time exists and has a clear direction. Evolution is the only physical process in our universe that can generate the succession of novel objects we associate to life. Things like microbes, mammals, trees, and even cell phones.
Well, that's an interesting concept. If time could go backwards, then and you still had novelty in evolution, then your descendants could each your ancestors, and therefore you would not exist.
Yeah.
Okay.
Yeah. So yeah, and then then they talk about information, memory. Such objects cannot fluctuate into existence spontaneously. They require a memory. So this is where you get into some sort of memory, and of course.
Okay, that first sentence the physicists argue with that.
Oh well, yeah.
Boltzmann brain.
Yes. So that
okay.
Yeah, originates spontaneously.
Yes.
Yeah. But
even in the universe, all things are possible.
Right.
Therefore, Boltzmann brains can come into existence spontaneously.
I think what they're getting at with this memory part is that life needs like something like RNA or DNA to have a memory of what came in the previous generation. So you pass on genes, or you have RNA molecules in there. You know, they get modified over time, where they're the same ones, and that tells the cell or the organism how to, you know, what to do. And so that's the idea of memory. Of course, we also have, you know, material memory, shape memory, biological memories, such as like, you know, cognitive memory or something like that.
Yeah, and the other thing I'm adding to that is we also have positive feedbacks.
Yeah. Is that a type of memory in and of itself, or is it like?
No, that's my model of. See, if you look at amphiphiles, which can form vesicles coming from meteorites, they're half as thick as modern membranes.
Right.
Okay, so the membrane had to grow in thickness over time somehow.
Mm-hmm.
And the way I postulated that is that if you have a process that makes peptides, even random ones, it will sometimes make hydrophobic peptides, which can go into the membrane and sort of stretch it a little bit, giving a bias towards membrane components that are bigger. So you get a positive feedback between the growth of peptides and the thickness of the membrane.
Standard definition of positive feedback is an explosion.
Right, but this saturates at some point for some reason having to do with the physical properties of membranes beyond a certain thickness.
Okay.
They seem to saturate at the thickness of modern membranes. This is not a well-explored area, but there are some properties that seem to have that they saturate, and also with temperature. For instance, the property is saturated at 100 degrees, coincidentally.
Yeah.
Okay, so yes, you can have an explosion, but if the explosion is limited by some other process, then it doesn't work forever.
Yes, hopefully.
Well, when you say positive feedback as an explosion, like what is? I've heard that term before.
Yeah, well, consider it a stick of dynamite. You light
Yeah.
it. Okay, the little bit of dynamite that gets lit burns, and it causes more dynamite to burn until the whole thing is burning and blows up.
Okay.
That's
So-
positive feedback.
But that would kind of be like runaway feedback.
Yeah,
Or-
that's a
yeah.
runaway
Okay.
positive
Yeah.
feedback.
You don't want that in a chemical plant.
Yeah, this is
Right.
what the communities like to hypothesize about carbon dioxide on Earth.
Okay.
Positive feedback, and the tipping point is when the explosion starts.
Yeah.
Our bodies get into positive feedback situations. For instance, in birth, that's positive feedback, but you can also bleed to death, I believe, if you get the wrong conditions going.
Yeah, there was a little book written in the 1960s on positive feedback. I don't know if anything's been done since then.
It's well studied in control theory.
On control theory, okay.
Yeah, it's
This
control
would be
theory.
before control theory.
Yeah.
There's lots of control theory out there.
Yeah,
Oh yeah.
yeah, but even in control theory, I presume that things can saturate, so you get a limit to how big the explosion is.
Right.
Well, yeah. Well, the chemical plant only has so much chemical in it,
Yeah,
and after exploding a couple of times, it's done.
yeah. And if something falls down, it can't fall down twice.
Yeah,
Right.
something like that.
Yeah. So they talk about so information and memory kind of gets into some of the this retaining the information and things like that. So
Okay, I argue with the first statement.
yeah, you did.
Again, with a small system, well, including in our paper, you can occasionally get a small system that goes through so many steps that it looks like it's had come into existence spontaneously.
Yeah.
And that's because if you have enough small systems, at least one of them might go through many steps and therefore come into existence spontaneously.
Yeah.
Okay, so this is catechism.
Right. Yeah. So to explain life, we therefore need to understand how the complex objects life creates exists in time. With her collaborators, we've been doing just that in a newly proposed theory of physics called assembly theory. So assembly theory is that as objects become more complex, the number of unique parts that make it up increases, and so does the need for local memory to store how to assemble the object from its unique parts. So that's basically maybe like our vesicles, where we have more particles or more particle types. This increases as maybe the object grows or becomes more complex. So then you need a local memory to store an assembly, some instructions for assembly. So if you have an organism that has maybe a couple of segments,
Bradley, can you put the URL for this in the chat?
Yeah. Well, I have it in the drive, so I can get the URL and put it in the, or I can send in an email.
Okay.
Actually, I should be able to get it here on the bottom of this page. Oh, I can't. Yeah, I'll send in an email after the
Thanks.
meeting. Yeah. So this kind of talks about assembly theory. They've done measurements on this in the lab. How a molecule with an assembly index of about 15 steps are only found in living samples. So this is like where they're assembling molecule or things. They're assembling macromolecules, I guess. And the more steps it has, the more likely it is to be alive. So this suggests that assembly theory is indeed capable of testing the hypothesis that life is the only physics that generates complex objects, and we can do so by identifying those objects that are so complex, the only physical mechanism to form them is evolution. So
Okay,
oh,
question.
Yeah.
Is a dendritic crystal complex?
I guess.
Okay, if it is, then it's generated spontaneously, and it has a complex structure, but it has no memory.
Yeah. Yeah, it gets into some gray areas here, but I guess for the purposes of their experiment.
Okay.
So this is the paper here, Algorithmic Origins of Life. So I'll send this along with that other article, but this is the actual paper here, Royal Society interface, and they talk about algorithmic origins. So that's the way they're selling it.
Yeah. Dave, I was at a meeting once where Davey showed up for a panel at the end of the meeting.
Yeah.
He's what I call a hit and run scientist. He comes to gives his opinion and then disappears. You don't get to talk to him.
Yeah. So they actually have some interesting things here. So although it has been notoriously difficult to pin down precisely what is it that makes life so distinct and remarkable, there's general agreement that its informational aspect is one key property, perhaps the key property. The unique informational narrative of living systems suggests that life may be characterized by context-dependent causal influences, or this top-down causation or downward causation. So this is where higher levels constrain the dynamics of lower levels. So basically, as you get more complex, things form these sort of hierarchical levels. The higher levels are constraining the lower levels. So this may be a major contributor to the hierarchical structure of living systems. Here we propose that the emergence of life may correspond to a physical transition associated with a shift in the causal structure, where information gains direct and context-dependent causal efficacy over the matter in which it is instantiated. So this is where it's, I guess, where information drives this sort of living aspect of the system.
They should observe some embryos.
Yeah. So they make the point that this is like a thermodynamic phase transition. So it's like a transition from one state to another, with some energetic input. So there's like a tipping point, and then there's a transition.
Yeah, that's exactly what's in the paper we're trying to get published.
Yeah.
There's a tipping point. We just assume it exists.
Yeah. So we discussed some, where the crucial distinction.
Maybe they should be cited.
Yeah, I think so. Yeah, definitely. Yeah. Where the crucial distinction that determining which phase, non-life or life, of a given system is in requires dynamical information, and therefore can only be inferred by identifying causal architecture. So that's like just, I guess, the history of the system, how it's interacting and so forth. So they actually do, I think, some experiments in this paper. They're going through the things we've talked about here. There's a digital origin of life. So they talk about the genetics first paradigm, identifying a digital information repository for living systems. So any living system has to have sort of a digital information store, whether that be DNA or RNA. And then a widely accepted resolution to this this genotype phenotype problem at the origin of life is the modern DNA protein world, where you've evolved from simpler precursor systems involving only one major molecular species that played both the role of information carrier and of enzymatic catalyst. So you have this sequence or this molecular species that was able to carry information and become an enzyme as well. So they talk about RNA being this mediator, and how it there's this.
Maybe it's unique.
Yeah.
I can remember the days when all enzymes were proteins, and therefore proteins were unique.
Yeah.
So.
No nitric oxide allowed.
Yeah. So, yeah, they talk. All right, so let me see if they have any examples from the experiments here. They just kind of go over more detail. They don't have any figures, actually. They just kind of go through the details of the theory. So they have this.
It's similar to what we're saying. At some point, you reach a configuration and you say, and there was life.
Yeah, yeah. Now, they do have this hallmarks of life table. So this is their hallmarks of life. And of course, everyone proposes sort of a threshold for life. So that's not like new, but they talk about things like global organization, information as a causal agency, top-down causation, analog and digital information processing, laws and states co-evolve, dual hardware and software roles for a genetic material, non-trivial replication. I don't know what that is.
Non-trivial, okay.
Yeah.
Okay, let's take a drop of water and go through that list.
Yeah, okay.
Okay, put your list back up.
Yeah, it's right here.
A drop of water has global organization.
Yeah.
Presumably has no information. Okay. Top-down causation. Yes, if you dent a drop of water with, let's say, a hydrophobic probe, you'll get a dent in the drop, but the configuration of that will depend on the global organization of it. So that's top-down causation.
Yeah.
Okay. It doesn't do analog and digital information processing. Okay. Fine. Laws and states co-evolve. I don't know what that means.
Yeah.
What's a UC?
I'm not sure. Universal. I don't know.
Okay. Non-trivial replication. I don't know. How trivial is a drop breaking into two drops?
Probably not if you want to have, you're in a microfluidic context. Yeah.
Yeah, it depends on the definition of trivial.
Right.
Okay, which seems rather arbitrary. Okay, there are no instructions. Okay, so a drop of water is halfway through.
Yeah, I think that's true. I mean, that's the thing about like these definitions of life is that a lot of times things will satisfy almost everything except for maybe one or 2 points, and it's like, well, maybe it is living. I don't know. So.
Yes.
Yeah. I mean, to throw a wrench in the works, you could say that like life maybe, or that water is maybe would be living, but it's like part of a living system because we have this sort of symbiosis with other things. So like water is a major part of life because it's maybe close to that boundary.
Yeah,
but,
these are obviously physicists, not chemists.
Yeah.
Chemists would start at the top of the list. It's got water in it.
Yeah.
I read I'm reading an interesting paper on gravity.
Okay.
And the old concept of habitable zone around a star?
Yeah.
Okay, it's a temperature range. Well, if you're too close to the star, your liquid evaporates, and if you're too far, it freezes. Okay, so this guy did the calculation for different liquids. And get quite different results. Okay.
Yeah.
So, raising the question, okay, is water the essence of life or not?
Right. Yeah.
Okay, so all these things are subject to very careful scrutiny.
Right. So yeah, this is a pretty good paper that I think it's worth like citing and thinking about. It's really just about like a theory of life. So it may be they're linking it explicitly to information and computation.
Yeah.
So they talk
See,
about
in our paper we're making the assumption that a metabolism is sufficient to get life started.
Right.
Now, metabolism, its structure, I suppose you could call a memory, but certainly it's not a memory in the DNA RNA sense.
Right. Yeah.
Okay, so we're not presuming to go all the way to a hereditable structure.
Yeah.
And what we say, the thing's alive as soon as it's got a metabolism. So that's the concept of autocatalysis.
Right. So yeah, they talk about Turing machines, for example, and they talk about just this need for, and of course, like metabolism is its own sort of information and memory. It's sort of like a memory of previous states, or staying within a certain range of values, homeostasis and things like that. So it's a very different type of regulation. But I think the question maybe is like, what came first? Did that information have to come first to build the metabolic system, or can we just have a metabolic system in
chicken and egg problem?
Yeah. So that's great. And then that's that paper. And then this paper here is kind of a sort of a high, it's what is this? Competitive exclusion principle among synthetic non-biochemical protocells. So this is an interesting paper. It's coming up in terms of the idea of protocells and kind of what they, you can do experiments with these different protocells. So this is having to do with, putting things in a protocell and looking at the sort of reactions that you get. So you have here a one-pot synthesis of simple non-biochemical but functional protocell populations. Two populations emerge when you put them into this protocell, differing only in that one has an advantage for reproduction. The populations compete for food in a shared environment, with one dying out. Simple protocells display Darwin's struggle for existence without biochemistry. So these are...
okay, what was the title? So
this is a competitive exclusion principle among synthetic non-biochemical protocells. So these are like, I guess, different molecules that aren't like, organismal, I guess. And they have, they put them in the same protocell with amphiphils, and they do this. And there's food in their chemical opportunities for chemical reactions, and you end up with these different populations. So in the abundant food case, you shine green light on the two populations, and you get this sort of configuration around the food. In an abundant food, it goes to abundant food, so they start to they can coexist, I guess, is the point. And then when there's scarce food, the protocell with ZNTPP dominates. They consume the food. The protocell without ZNTPP dies off. There's just one little colony here, or this one little protocell here. And so the idea is that with abundant food, you can get coexistence of these two protocells. And then with food, and they don't really consume all the food. And then when there's scarce food, one dominates. So this basically says that, when you have a certain chemical composition in a protocell, it can outcompete other protocells. And so it's kind of an interesting study in these really early cells and what their contents are and how they started to sort of emerge as, maybe giving some of that memory and what it can do. So
did they actually do experiments?
Yeah, it looks like they did experiments with these protocells. So they put them in these different environments with the green light and with different amounts of food, and they show.
Okay.
Yeah. So the summary is essential for Darwin's struggle for existence. The competitive exclusion principle (CEP) states that any two species occupying the same niche will compete with each other to the detriment of one of the species, which will thus be excluded. That's why they're doing this kind of an experiment.
That concept is, by the way, sort of a symmetry breaking.
Yeah, it is. Here we report on competition experiments between two populations of autonomous artificial self-booting, self-reproducing polymer-based protocells. So these are where you're putting in like different elements in the protocell and letting them loose, emerging from a homogeneous blend of small synthetic chemicals in a one-pot reactor using polymerization-induced self-assembly, which is PISA. That's their definition of that. These protocells are carbon chemistry based, but biochemistry free. So you can do carbon chemistry, but not biochemistry. These populations share their environment, differing only in that one contains a photocatalyst that confers an advantage in reproduction. So that's what the green light was for in that figure. Competition is shared in the shared environment follows the CEP, competitive exclusion principle. Thus, biochemistry is sufficient but not necessary to drive the CEP. So we don't need biochemistry to have this sort of lifelike behavior, even like a Darwinian type of evolution.
Okay. Does their experiment fit Walker's conditions for being alive?
That's a good question. I'm not sure. I guess it has like a lot of these elements. I'm not sure which ones it's missing.
I mean, if it's self-reproducing, it doesn't need heredity.
Yeah. I mean, it's not. They're not like. I guess they're biochemicals, but they're not. Or they're compounds, but they're not biochemicals, or they're not the sort of DNA RNA paradigm which we're used to.
Yeah.
So I guess you could say, well, you don't really need that, but the question is like, okay, what? I guess it's just that it's following Darwinian principles because it's able to outcompete the others. So it'd be like if you have a competitive binding experiment.
Yeah,
Sometimes
so
they do
most people make the assumption that once you reach a Darwinian state, that you are alive, and therefore the problem of origin of life has been solved at that point.
Yeah.
Okay. So that's why I raise it with this thing.
Yeah. So I don't know which points they hit.
Yeah, it'd be interesting to take this paper and put it against their checklist.
Yeah. I'm going to say no because I don't think that it has all those elements. But that's
no, it
kind of the point.
It doesn't. But,
yeah.
So what?
Yeah. So they go through their methods here. They just kind of show.
For instance, suppose you could only eat the one that dies off.
Right.
Until you.
So I mean, to be clear on the chemistry here, you have chemical structures, but they're not encoding any sort of particular memory. Like DNA would be maybe expressed, or RNA would be expressed, or it could store information. This doesn't. This is just basically an identity. Yeah. And then, this is showing the sort of the different protocells with their components, and then it shows like how they're receptive to photosynthesis or photostimulation. So that's a key part of the experiment.
Yeah,
and
of course.
Then,
Go back up there a second to that porphyrin.
This one?
Yeah. Yes, that one.
Yeah.
That molecule looks suspiciously like something that's been synthesized by a living organism.
Yeah.
Okay, so they say non-biochemical, but what is this thing?
Yeah, I don't know.
Where'd they get it? Who sold it to them?
Yeah.
It's a Sigma chemical. Oh, we don't care where it comes from.
It's like a photosynthetic element, right? I mean, that's basically, I'm assuming it's very, yeah, it's
Yeah, yeah,
responding to
yeah.
light. So
Porphyrins, I don't know if any porphyrins are made spontaneously anywhere,
yeah,
other than living organisms.
right. So you have a starting point that's already structured for a living
Yes,
system. Yeah,
could be.
yeah,
In other words, the claim is that the system is non-biochemical may be false.
right, right. Depends on how you define biochemical, I guess.
Yes.
So yeah, and then this just shows these experiments where they show population growth with respect to the, the competition experiments over time, and yeah. So I think that's it. I don't think there's much more to the paper. Well, they show like polymerization and fast polymerization, slow polymerization. So they show these. This is basically how you get this differential population. In this population, there's fast polymerization. In this one, there's slow polymerization. So this one gets out competed because it doesn't have the ZNTPP molecule that we saw up above. So yeah, that's all. Okay, now I want to talk a little bit more about symmetry breaking and development. And this is a paper from the special issue Royal Society Interface Focus called Symmetry, Simplicity, Broken Symmetry, Complexity. So the idea here is that symmetry means that you start at a simple state. So symmetry is sort of the null condition. And then complexity emerges from broken symmetries. So if we think about that in terms of the embryo, or maybe some sort of symmetrical system, we can start maybe with a twofold symmetry, something like this, where you have two parts that are even, but they have a midline here, that can be defined by the squiggly line. And so that's the midline there. And then we could have fourfold symmetry, which would be something like this. I think in our paper we give an example of fourfold symmetry, although it's technically twofold symmetry in the C-squirt. So that's this actually, now we can go to, I've just drawn a fourfold system where we have the four lobes and these two axes. There. So that's a very simple way to sort of characterize this. I like this paper because it actually talks about emergence, and it talks about this paper by Philip Anderson, which is actually a good paper to think about complexity theory and the sum being greater than the parts. So this is an early paper on emergence, and this actually has a lot to do with development as well as just general complex systems here. So I wanted to also bring our attention to some of the symmetry breaking stuff that we've done in the past. So this folder here is has pattern formation models from August 9, 2021, symmetry transformations and metazoan evolution from May 31st, 2021, and this annual review of cell biology paper, which is new to us. So if we go back to pattern formation models, we had a couple papers in that in August 9th of 21. One paper we talked about is this old paper from 1940, and this is very old, but this is Reshevsky, and he talked about physiochemical mathematical aspects of some problems of organic form. So this is very, very early embryology and sort of the idea of symmetry and symmetry breaking. So if we go back to our board, we can see that we actually have a problem here whereby we start with a single sphere or something oblong perhaps, but we'll start with a sphere. And then we have these symmetry breaking events. So we can map development out as a set of symmetry breaking events. We might have something like this. So we have these we have, say, an anterior-posterior orientation of a single sphere. This would be one example. This would be another one. This would be a third. The symmetry breaking events then could proceed as, say, from this point, and you get a series of symmetry breaking events that maybe unfold like this. And you can see that what's happening here is that you have cell divisions that are occurring at different rates. So this is twofold symmetry breaking here. This is where you have an asymmetry between this cell and this cell. The cell's divided, the cell hasn't. This cell is divided twice, this cell is not only divided once. This cell is divided 4 times, and this cell is divided twice. You can see in each pole you're having this greater and greater asymmetry. And you can see how it contributes to complexity in different regions of the embryo. We can assume that this is A and P, and this gives us not only an asymmetrical embryo, but it starts to lead to different tissues forming and different organs. We did some work like this in one of our papers from 2016. So if you're interested, I can cite that paper for you, and you can look at it. So this is a nice starting point, this paper, and then this paper, of course, is also sort of the roots of this in embryology. We also talked about pattern formation for pneumatic liquid crystals modeling, analysis, and application. This was a little bit more on like soft active materials and some of the things going on with liquid crystals. And of course, symmetry is important in liquid crystal formation, so we talked a little bit about that. This is a paper on mathematicians prove symmetry of phase transitions. So this gets into the idea of phase transitions and how those are important for breaking symmetry. As you can see from this example, you're undergoing a phase transition when you have these asymmetries. So there's a change, a very quick change from one state to another. And you get like perhaps one part of the embryo undergoes a phase transition relative to the other part. And so you get these asymmetries. But this is a group of mathematicians have shown that at critical moments, a symmetry called rotational invariance is a universal property across many physical systems. And so this talks a little bit about rotational invariance. And rotational invariance is a symmetry exhibited by the circle, rotated in any number of degrees, and it looks the same. So this is where we're looking at the circle from different orientations. We're considering it from different orientations. And that's actually something you have in the embryo. I'm showing you a two-dimensional sort of flat surface, but the embryo is a rotational thing. It's maybe oblong, it's maybe more spherical, or it's maybe differentiated a bit, but it's this three-dimensional shape. And so when we rotate it, we can get different perspectives on it, but we also have different things going on at different orientations. And so that leads us to this last paper here, which is the dynamics of morphogenesis and stem cell-based embryology, novel insights for symmetry breaking. This is from 2021 in Developmental Biology. The abstract of this reads, breaking an embryonic symmetry is an essential prerequisite to shape the initially symmetric embryo into a highly organized body plan that serves as the blueprint of the adult organism. So what we were just talking about with the images. This critical process is driven by morphogen signaling gradients that instruct the anterior-posterior axis specification. Despite its fundamental importance, what triggers symmetry breaking and how the signal gradients are established in time and space in the mammalian embryo remain largely unknown. Stem cell-based in vitro models of embryogenesis offer an unprecedented opportunity to quantitatively dissect the multiple physical and molecular processes that shape the mammalian embryo. So in this paper, they review biochemical mechanisms governing early mammalian patterning. So we were kind of using a C. elegans template here, but we could also look at mammalian patterning as well. And to recreate this in vitro using stem cells. We discuss how the novel insights from these model systems extend previously proposed concepts to illuminate the extent to which embryonic cells have the intrinsic capability to generate specific, reproducible patterns during embryogenesis. So they're using this special type of system to look at embryos and how they might do symmetry breaking, and they're looking at this problem of antero-posterior axis specification. So they're trying to get these cells to differentiate in different locations and break the symmetry of sameness in the early embryo. So this first part might be useful here. During gastrulation, for example, which is a stage of development, the entire embryo undergoes coordinated morphogenesis to establish the future body plan, while seemingly relying on sophisticated interactions within and between cell tissues. This process requires complex networks of signaling interactions at the level of single cells, as well as geometric and topological transformations at the level of the embryo. So we've talked about this with respect to this little chart here. You have these topological changes, but within this you have signaling differences. So there are signaling differences that are going on in the anterior and the posterior end, and as those differentiate, they lead to this phase transition. It should be a sudden change, maybe corresponding with some sort of cell division, but you still have this aspect of sort of multi-level or multi-scale symmetry breaking. So changes at the smaller scale contribute to these bigger changes at the higher scale. So molecular changes contribute to the cellular phase transition, and you get symmetry breaking. This complexity is amplified at gastrulation. So at this stage of gastrulation, where embryonic radial symmetry is broken to establish the anterior-posterior axis and germ layers, these are subsequently specified. So we have germ layers, the AP axis. This is in the mammalian embryo. We have this radial symmetry in the early embryo. And in mammalian embryos, you get like a fair number of cells that are these, totipotent stem cells that are in this ball, and then eventually they start to differentiate into an anterior and posterior end, and they provide this in germ layer, in the 3 germ layers. So how the early embryo can undertake this complex transformation with such fidelity and precision has remained for the most part a mystery. This is primarily because decoupling the cellular movements in different parts of the developing embryo is prohibitively difficult. So this is why they're using the stem cell model. So this paper, we talked about this back in 21. This shows kind of the idea here. You have this pre-implantation embryo, you get a pregastrula, an early gastrula, and then you get the morphogen expressions which shift in different regions of the embryo. Now this is the interaction between molecules and cells. We actually talk a little bit about symmetry breaking in the context of some of the work that we're doing in our paper. We're talking more about tensegrity networks, and so that might be worth reading our work in the special issue here, this paper. So in this paper we talk about various symmetry breaking events in developmental time. We talk about the 4E cognition concept: embodied, inactive, embedded, and extended. And so in this context of these different forms of cognition or non-neuronal cognition, we talk about symmetry breaking. We talk about how tensegrity networks might play a role in that, as well as other things as well. So it's very, it might be quite interesting for people who are interested in this topic to take a look at. So the next thing I want to talk about is this paper, Symmetry Transformations in Metazoan Evolution and Development. This paper is from the journal Symmetry, so fitting. The abstract says, in this review, we consider transformations of axial symmetry in metazoan evolution and development, the genetic basis, and phenotypic expressions of different axial body plans, in addition to the main symmetry types in metazoan body plans, such as rotation, which is radial symmetry, reflection, mirror and glide reflection symmetry, in translation, metamerism. Many biological objects show scale fractal symmetry, as well as some symmetry type combinations. So this is something where they're talking about this three-dimensional volume where there's this rotational invariance, and you have these different aspects of symmetry. So in rotation, you have radial symmetry, reflection, you have mirror and glide reflection symmetry, and in translation, you have metamerism. The idea is that as the embryo changes, there are a lot of complex topological changes, not just something as simple as this, which can symmetry break in these different ways. So you can get symmetry breaking in rotation, in reflection, in translation. So these are all different aspects of symmetry breaking, which are topologically complex. And they really start to show up as the embryo starts to differentiate. So, this paper, lost body plans of some extinct Ediacaran and early Cambrian metazoans are also considered in comparison with axial body plans, posterior growth in living animals. So this is a paper where they're using like early embryos, you'd call them, and comparing them with modern embryos or modern organisms. And of course, they're comparing the developmental aspects of them. So this is relevant to what we've been talking about recently with early embryos and things like that. So, but I think what they're really trying to do is get at some of these aspects of topological symmetry breaking. So there's helicoidal symmetry, which involves the nodal pathway. Helical spiral symmetry involves rotation combined with translation along the axis of rotation. So this is where these two things, along with spiral symmetry, rotation, and translation occur together with an expansion. Each rotation is order as a number of repetition of parts that are arranged around a full circle. So you get changes in this as things are rotated around a full circle. You get changes in the number, and that's the symmetry breaking that you're going to observe. So in Spiralia, which includes annelids, mollusks, and flatworms, spiral cleavage produces a highly stereotypic spiral-like arrangement of blastomeres and swimming trochophore-type larvae with rotational symmetry. Mosaic development, where most blastomeres inherit distinct maternal determinants, is also considered characteristic for the Spiralia. So mosaic development is like what we see in C. elegans, where there's a defined lineage tree and the cell fates are deterministic for the most part. So this kind of goes through this aspect of, this is just one example of symmetry and symmetry breaking that you see across life, in the tree of life. So in this figure, you can see that there is a fractal colonial organization of barnacles. And so you can see that there is symmetry and asymmetry in this as well. This is a spinal neuron. This is ontogenetic changes in morphology and fractal dimensions. So as spinal neurons mature with their processes, they change over time and they undergo this sort of symmetry breaking in the fractal dimension. So it's a little bit more complicated than like just simple, what you might call linear symmetry breaking. Okay, so that's, and then finally, this paper, Symmetry Breaking the Mammalian Embryo. This is a nice review of what we know about symmetry breaking the mammalian embryo. The abstract reads, we present an overview of symmetry breaking in early mammalian development as a continuous process from compaction to specification of the body axes. While earlier studies have focused on individual symmetry breaking events, recent advances enable us to explore progressive symmetry breaking during early mammalian development. Although we can primarily discuss embryonic development of the mouse, as it is the best studied mammalian model system to date, we also highlight the shared and distinct aspects between different mammalian species. Finally, we discuss how insights gained from studying mammalian development can be generalized in light of self-organization principles. With this review, we hope to highlight new perspectives in studying symmetry breaking and self-organization in multicellular systems. So this is a really nice reference. They have, they have a table of contents. It's not that long of a paper, but they talk about the first symmetry breaking event, symmetry breaking in the blastocyst, the embryonic body axes, which we've talked about, the insights we can gain, and future perspectives. So the first symmetry breaking event, and so in many cases of animal development, symmetry breaking occurs continuously from sperm entry to body axis establishment and germ layer formation by gastrulation. In mammals, this process spans pre-implantation development in the oviduct and uterus, implantation and post-implantation development in the uterine tissue. So symmetry breaking occurs really early, according to them, and you see this even from sperm entry into the ovum. And so this is where you get just the series of symmetry breaking events from a very early point onward. And so to study symmetry breaking, it is also worth critically evaluating the presence of symmetry, namely whether the initial cell population is equivalent by which definition. So in order to know what the parameters of symmetry breaking are, you really need to know what symmetry is when you're talking about this. And sometimes it's not obvious what symmetry is. As we said, there are many mathematical definitions of symmetry. There are many topological definitions, but there are also these very simple definitions, which could be axial, and they could be very straightforward. So the first symmetry breaking event, so there's been considerable debate, actually, to when symmetry is first broken between cells in the early mammalian embryos. And so it looks like there's a lot of stuff in here that Zernica gets, Yamanaka, there's some pretty decent names in here where people were debating this. So the root of confusion primarily stems from the fact that mechanisms of symmetry breaking and embryonic axis formation do not appear to be conserved between mammalian and non-mammalian model organisms. So if you look at Drosophila, C. elegans, Xenopus laevis, those are all, they don't really share any traits of the mammalian embryo in terms of symmetry breaking. Such lack of conservation is evident, for instance, from the failure to identify and functionally validate so-called maternal determinants in oocytes and potentially early blastomeres. So furthermore, although possible roles for sperm entry and asymmetry between two cell stage blastomeres, so we're talking very early on in embryology, have been proposed, deterministic mechanisms comparable to those non-mammalian species have been established in mice. And so, this deterministic mechanism means that it's something that is almost like programmed into the embryo and should happen at a specific time and in a specific way. More recently, possibly symmetry in terms of difference in gene expression, transcription factor dynamics, and developmental potential among 4-cell stage blastomeres was proposed. And so this was proposed by a number of people. However, other studies were unable to substantiate this claim in terms of gene expression. And so this is a problem where you have different types of evidence telling you different things. Cleavage patterns, cell lineage, and cortical tension. So these are different aspects of cell lineage, of course, and cleavage pattern have to do with the cell lineage tree, and then cortical tension is a property of the cell itself. Given the non-stereotypical division pattern and dynamic rearrangement of the blastomere and polar body upon cleavage, it is crucial to develop methods unequivocally identify the individual blastomere of the 4-cell stage embryo for future studies. So this kind of goes through, let's see, figure 1 actually shows some of this. So these are overview of symmetry breaking events. So some of these are symmetry breaking events that have been defined, but we're trying to define the first one. So this is the zygote. You get a 2-cell stage, you get this 8-cell stage, and then the compacted 8-cell stage. And this is one of the first symmetry breaking events because you start to get this layering. So at E2.5, this is in mouse, you get this compaction. So this compaction leads to an inner layer. And this is the apical domain out here. This is the inner layers, lineage specification, where you get further differentiation by the differential gene expression And then that all happens within about a half a day. And then E3-5, you get a blastocyst, E4-5, a blastocyst, and then you get to the pregastrial stage. So you see maybe this is the first symmetry breaking event. Some people, I guess, think that it's back here at the zygote. And so this is where the debate lies. So this is these are two models. There's the inside-outside model, where you get this outside and inside symmetry breaking. And then you have the cell polarity model, where you get polar and apolar cells. This is an asymmetric division. And then symmetric division, where you get two polar cells. In this is the region we're talking about. So this is a symmetric division. This is an asymmetric division for the cell polarity model. This is the inside-outside model. So the inside-outside model hypothesizes that cells on the outside of the morule are exposed to a different environmental and therefore different stimuli relative to cells on the inside of the morule. Due to these environmental differences, outside cells develop into trophectoderm cells, while you also get an inner cell mass on the inside. The cell polarity model proposes that with the acquisition of apical basal polarity at the 8-cell stage, the angle of cell division dictates cell fate. Asymmetric divisions, where the plane of the division is perpendicular to the axis of polarity, segregate fate determinants differently between the daughter cells, leading to one adopting the TE fate and the other adopting the ICM fate. So you get this. I think we also talked about this in this 2016 paper I'm referencing from the group, where you get this cell division events where things segregate asymmetrically within the daughter cells. So you get this, different factors in different cells, and they end up in different places, and then they end up forming the inside and outside. So it's not just like a signal that says these cells are going to the outside, these cells are going to the inside. It's just the factors that get segregated during cell division that determine this. And so you can see that they're asymmetric divisions and symmetric divisions. And this gives us a model for some of these changes. Conversely, a symmetric division parallel to the axis of apical basal polarity would yield two cells of similar fate. So, yes, and then we have cell position, apical domain orientation of cell division, cortical tension contractility. This is their model for symmetry breaking. So this is the apical domain reconciling the inside-outside model and cell polarity model. So you see that you can have the cell position as sort of the place where you start. You get this apical domain. You either go down this path of this cortical tension and contractility aspect, or the orientation of cell division, and those both can lead to asymmetry, and mechanosensing and apical basal polarity sensing also lead to this differential aspect. So that's another version of asymmetry. So you can see that early on in the embryo, you're getting this interaction of physics, molecular changes, and spatial orientation, and they're all playing a role in different symmetry breaking events. So I think that's good enough for now. I think the rest of the paper talks about a specific case of symmetry breaking in the blastocyst. And then if you go down to the third part, actually this is kind of showing this in a figure, spatial segregation of primitive endoderm. So you can see that, again, space plays a big role in this. And you have apoptosis for certain cells, you have fate switching for other cells, so you get changes going on like that. And then you get this, then the insights we're gaining from this. So early mammalian embryos provide an excellent model system in which to study the multicellular symmetry breaking and self-organization. It's a relatively simple system, but complicated enough to look at some of these issues. And so the principle of self-organization may be conserved across species, even in those embryos classically considered to adopt a deterministic developmental program, so like C. elegans and Drosophila. So this is actually something that really is, it sets these two kinds of systems apart. So you see this sort of deterministic developmental program in mammals, but generally mammals have this regulative capacity for embryos. So this is why, we have to kind of focus in on some of these more complex processes. Okay, and so that's, I think, all for this topic. Thank you for following along, and hope you learned something. All right. Well, that's all for today. I'll send those out in an email. Some of the things. Yeah. All right. Thanks a lot.
Bye.
Okay, thanks.
Talk to
Bye.
you later. Bye.